Hello everyone, welcome to topic 3.9, separation of solutions and mixtures chromatography. In this we will look at how we can explain the relationship between the solubility of ionic and molecular compounds in aqueous and non-aqueous solvents and the intermolecular attractions or interactions between particles and how we can use that to separate mixtures. So, starting with our notes, chromatography provides a method of separating a mixture of solutions based upon polarity differences. The polarity differences are sometimes considered solubility as well. There are three types of chromatography: paper chromatography, thin layer chromatography, and column chromatography. We will do paper chromatography in class. In general, a line is drawn, usually with a pencil because it does not move along the paper near the end of the strip. It is placed into a sample, solvent, and, a sample is placed on it, and then it is placed into a solvent and sealed. The solvent then climbs up the chromatography paper and separates out the colors of the dyes in the sample. And then you remove it before it reaches the top and mark the band at the top to indicate where it reached and how far it made it. You can then measure the heights and compare rate of flow and connect these to the polarity of the compounds in the mixture that you spotted for your sample. So paper chromatography analysis, the same sample will move at the same rate relative to the same solvent on different trials. The different compounds will have at least slightly different polarity to other compounds, so the rate of flow, or RF factor, that is what that is called, is rate of flow. So how fast it flows through the paper, will differ between compounds. And that is a fraction based off of how far it traveled and how much the total distance was here. And since you're always comparing those two and it's a ratio, you can compare them across other trials as well, and it should be similar. The more similarity in polarity to the solvent, the farther it travels. Polar water will have polar samples travel far, and nonpolar samples will travel only a short distance. Nonpolar benzene will have polar travels travel only a short distance, so not very far, and nonpolar samples will travel far. So basically, the sample will travel based off of how much it likes the solvent or elute, the solution that you're using to put the paper in. It'll travel based off of how similar its intermolecular forces are to it. Okay, you identify the sample by comparing RF values. You never compare direct distances because the solvent front will travel different distances. And you can identify samples from that. The stationary phase is always the paper, and the mobile phase is the solvent. In the image, the mixture doesn't contain dye 2, as you can see, because it's not there. It does contain both dye 1 and dye 3. It is not uncommon for dyes to have multiple parts in which it would look like multiple dots, different distances from the baseline. We are going to do that with food dyes in a candy chromatography lab in class. Thin layer chromatography is extremely similar to paper chromatography, except that the separation occurs on a thin sheet of plastic. The layers typically silica or alumina, which are extremely polar. Thin layer chromatography can be used to separate samples that are not colored to the naked eye. You can use UV light with a solvent that fluoresces under UV light to see the substances. The dark spots are where the components of the mixture are. This method is frequently used with amino acids, so protein synthesis or protein analysis. So expect this topic when the question also refers to biological systems. Thin layer chromatography analysis, the same compound will move the same rate relative to solvent on different trials. So that's similar to paper chromatography. Different compounds will have at least a slightly different polarity to other compounds, so the RF factor will separate or differ between different compounds. The more nonpolar the sample is, the farther it travels, because it's not very attracted to your plastic sheet or your stationary phase. The more polar the sample is, the less it will travel. The leading edge must be identified and not allowed to reach the top, just like in paper chromatography. And identification of a sample is used by comparing RF values. Again, not distances directly. Column chromatography is one you may see when you take college level chemistry classes beyond AP chemistry. This separates, especially organic chemistry. This separates a mixture in solution, but this will separate them out so you can reuse the separate components. So something is packed into the column to prevent to help separate it out. The gel is in here, and then steel wool as well to prevent it all from flowing through. A burette is filled with polar silica or alumina gel, just like thin layer, but this is a column to allow the solution mixture run through. The mixture is to be separated, is placed at the top of the burette or whatever column and flushed through the column. And then it'll separate based off of its intermolecular forces. So non-polar solutions that are poured in at the top will go through faster. So polar travels the slowest, and least polar will travel the fastest. Once you have separated the parts, you can use a new solvent to speed up the movement of whatever's remaining. So like water or something else. Okay, this is meant for separation more than analysis. So if you have a mixture of two and you want to do further analysis beyond just seeing if there's two or more things in there, you would separate it using this and then run further analysis. Another way to separate mixtures is distillation. This separates substances based on differences in boiling point and is thus tied to intermolecular forces. There is a distillation apparatus shown to the right, and it walks you through. But basically, something is heated here. The one with the lowest boiling point will boil off first and come up. It will then be cooled in this jacketed water condenser. Water is continuously flowing to help cool it down, and then it is collected in a separate container. There will be a break when in something boiling and coming off, when all of the first compound or the next one, when all of the one compound that was boiling is off, and so you can then switch your collection container and collect the next one. There are a lot of applications, including distillation of fermented beverages, desalination processes, separation of crude oil into fuels and other petroleum products. That's fractional distillation. Typically, the boiling points are close together. If the boiling points are close together, fractional distillation is better because there is repetitive cooling and condensing columns as you go up, and so it allows things to separate a little bit better. So if we look here, it wants us to identify how many dyes on this paper chromatography. So when we look at this, we see we have ones all the way up here at the top. So there is one. And then we have these two yellow bands here. So that is our second. We have this pink band that probably goes, well, not that one. That pink band, I would call that my third. There's this one that might be a fourth, or this one might be over here, and this is a separate one. And then we have this group here as a fifth. So we should see 5 dyes. This fifth one might be part of this, or it might be tied to this one. You would actually have to compare the rate of flow values for this. Okay. Next is our redo. Serine and glycine are both amino acids where serine is polar. Serine and glycine. Serine is polar and glycine is nonpolar. The two amino acids are not labeled and must be identified. Explain why thin layer chromatography is more appropriate than paper chromatography in this case. So for this, they are both colorless. And can't be seen on paper. You would use UV lights to have them fluoresce. I may have misspelled that, but whatever. Describe how to set up a thin layer chromatography experiment that would help to identify the two amino acids. Well, I would say plate the mixture and known reference samples. If not mixed, so we don't have a mixture, just two unlabeled bottles. And then you would run it according to the thin layer chromatography. So draw the chromatogram that would result. So if I'm assuming that they are mixed together, I would draw it like this, right? And I'd have my origin spot for my unknown, and then I'd have my serine, and then I'd have my glycine. And I would expect serine being polar not to travel very far, and then glycine to travel really far. And so I would expect then my unknown to have two. If they weren't mixed, I would then have 4 spots with serine and glycine and my unknown 1 and 2, and I would expect to see some combination of this where one of the unknowns matched glycine and one of the unknowns matched serine. But of course you would do an RF calculation and compare those. Okay. From here you are going to complete the you-do using conceptual information and a little bit of math to compare and dive deeper into chromatography. Thank you guys.